Hello. How are you guys all doing? I see that some of you are already here as well, which is exciting. Look at that. And as you've been told to be civilized, so make sure that you are civilized. How are you guys all doing? Hey, Angela, how are you? Anyone else? Just Angela? Angela's all the only person who's here, yeah? I think it says on my thing there are at least 43 of you. Zoe, hey Zoe. Hey Jaden. Finally. Hey Rose, or Jasmine, sorry. Hey Daniel. Daniel, that is the most interesting way I've seen Daniel as well. Nice. Hey Phoebe, hey Zara. Azika, football highlights, how you doing? Any of you caught up in Storm Dennis at the moment? I was intrigued as to if anyone would actually be in Wales at the moment and like basically drowning. Hey Ria. Hey Samia, hey Himaria. Hey. Let's have it. Good shouts. Yeah. So as Ola, as part of our I don't know what it is, like tricky topics in biology, hopefully some of you guys responded to the Instagram thing a little while ago. The plan is to basically go over things that you guys don't like very much. So I did photosynthesis last week. Today we're going to do respiration, or I did the kidneys, I think, before that. And then I guess we'll have to see how you guys are feeling in terms of what you want next, in terms of, I don't know, whatever's hard. People don't like inheritance. That's normally quite hard, but I'll see what everyone's saying. Oh, good. I'm glad that at least one of you is getting a batter in terms of Storm Dennis. Storm Cara was worse for me as well in the southeast. I think it's like Wales, isn't it? And like going over to Ireland where it's starting to get really bad for Dennis. Anyway, genetics is nasty, so maybe that. Yeah. People don't like that traditionally. Okay, genetics could be the one. I might make this tomorrow then. Right, let's get cracking then. So I thought that I'd try and cover pretty much all of respiration because most people tend to do like one of a few things, right? So if you've never come across glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation, today is going to be a little bit hardcore, so I can only apologize for that. But if you have done this, hopefully this is something which is going to summarize everything. PCR and that stuff as well, Zoe, I totally agree. I want to go over that at some point, but possibly a bit later in the year, just because I know that lots of people won't have actually done that yet. So I don't want to go through that when like two people have done it. That seems a bit pointless. Exactly, unicorn power, exactly. So in terms of spec points then, here are AQA, and AQA basically has everything written out in loads of detail, so you can come back and have a look at this at some point. I'm not going to bother reading it. There's OCR. Same kind of thing, quite a lot of detail. EdXLA, that is a lot of detail actually. So feel free to come back to this. It'll be on YouTube when I'm done. Have a look, see what you need to know, and just literally tick off the things that you think are important. And then EdXLB being insane here of just everything, literally everything here that you need to know. Oh, amazing, Daniel. If you can do that, that'd be very useful. Do I cover any EduClass exam board? I've never even heard of that. EduClass. If it's similar to EdXL, which it sounds like it might be, then yes, but otherwise I don't know. I guess you'll see. You have to wait and see. It'll be a surprise. Okay, so in terms of respiration, then, I need to turn my laptop on silent. There you go, much better. So in terms of respiration, I think it's important that we sort of begin by working out where everything actually happens. So in terms of the mitochondria, then, does anyone know what the names of the different parts are? So if I've got three main sections, what are the names of the different parts? Who can tell me that? The matrix, cool. Which one of these is a matrix? Cool, Grist A is also good. Not to be confused with Cystine A, which I'll tell you about in a second. That one doesn't help. Hang on. So let's go 1, 2, and 3. Which one am I looking for the matrix? So yeah, good. So 1 is the matrix, totally agree. So it's kind of like a cytoplasm, but it's got a load of chemicals in it which are in a much greater concentration, like TP and like pyruvate and stuff like that. T, what is T? Unicorn power. Bear with me, I'll tell you in a second. What is T? What am I talking about here? Membrane. Be more specific. Good. That's the outer membrane. And 3. Be careful. It's not a cell membrane, number 2, because this isn't a cell. It's an organelle. I know it was once a cell for those of you who are about to tell me that, but it's not anymore. Good. So these are the cristae. Okay. So the cristae being the things which are covered in enzymes like ATP synthase and I don't know, some electron transport carriers and stuff like that. Okay, cool. So whereabouts then? Whereabouts does glycolysis occur? I'm being a little bit harsh here, but where does glycolysis occur? Can you tell me? Cristae are folds of the matrix. Daniel, be careful with that as well. The matrix is the liquid part. The cristae are the membrane bits. Good. So well done for remembering this. Glycolysis specifically happens in the cytoplasm. Most people forget that. So this is why when you're told at like key stage 3 and key stage 4 about what happens in the cytoplasm of the cell, you're told that that's where reactions go on. That's because it's where glycolysis goes on. Okay, link reaction, where's that going on? Good. So this is a mitochondrial matrix, totally agree. Krebs. Yeah, Krebs is the same. And oxidative phosphorylation. This is difficult to actually, well, feasibly difficult to say. I guess not particularly. What do you reckon? Yeah, so the inner membrane space, the cristae, something along those lines. So depending on what you got asked, if you got asked where does the chemiosmotic gradient of hydrogen ions, where does that occur? You'd want to say like the inner membrane space. If they said where does ATP get produced, then you could say it's the cristae. If they asked where the electron transport chain is, I would say the cristae again. Okay, so I'm going to write cristae for this. So this is a really standard A-level exam question of just where specifically do things happen, right? And you've got to get out of the habit of just saying the mitochondria. You need to start thinking of this as being like a specific place where each of these actually happened. Just because someone said what is a cristae as well, you might have come across this. And I might have spelled that wrong. It might be an E there, but I can't remember. No, it's cystine, isn't it? Oh, totally losing my plot. The cystine, which is cyst. Oh, I have definitely spelled this wrong. Bam. The cystine are essentially the membranes that you find in your Golgi and in your endoplasmic reticulum. And it's something that some of you are expected to know. So be careful about cystine because people confuse it with cristae quite commonly. But anyway, let's carry on going. So that's like the overall kind of respiration. I guess you guys probably all know what the equation is as well. For aerobic respiration specifically, you guys need to know that you're making glucose, you're using glucose, so you're reacting glucose with oxygen, and you make carbon dioxide and water, and you should know that you have 6 of all of them apart from glucose, which is C6H12O6. But anyway, if we look at glycolysis then, glycolysis is where this starts to get a little bit complicated. And I think one of the things that people slip up on is they get confused as to whether you are making ATP and if you're making reduced NAD or if you're using them, right? People tend to get really kind of caught up on this. So I would try to remember this as ultimately, you should all know that it's this guy here, which is used to make loads of ATP at the end. So all of respiration, it's all about making reduced NAD. That's like the most important part, with ATP being a bit important and FAD being a bit important too. Awesome. Right, let's have a look at this then. So if we start off then, the very first step, and I've got 4 bullet points here, so let's go through it like this. The very first step is I am doing something to glucose. So some of you might remember this, some of you might not, but you actually need to activate glucose first. And this is quite a standard thing that we do in biology, where you give a phosphate group from ATP, well, there it is, from ATP, you give that phosphate group to another molecule. So the very first step is you add some phosphate groups to glucose, and it will either make, and it depends on your spec here, something called hexose bisphosphate, or it will make something called glucose phosphate. Depending on what spec you do, it'll be one of those or the other. Okay, so the very, very first step is we need to know that glucose is phosphorylated, which uses 2 ATP molecules. Okay, yeah. So with this sort of thing, you don't need to have this perfectly off by heart forever, but I've seen loads of questions where it will say like, name what this molecule is here, right? Or name what this molecule is here. So you need to have a good enough understanding of this. So I would just write this out enough times until you know it pretty much like as well as you feasibly ever could, right? So you've got to get this sort of in your head. Does the phosphorylation of it make it polar? If it's got a phosphate group, phosphate is charged. So yeah, I suppose it does. It doesn't last very long. It splits in half very quickly. What else are we saying? Aisha, what are you talking about? You're just talking about the diagram. Oh no, you're talking about Zoe's question. Glucose is not reactive. So yes, Zoe, no, you are spot on. So by adding a phosphate group, you are helping this reaction to happen. You're sort of giving it a bit of activation energy so that it can then carry on. So you are spot on that you need to activate your glucose by adding a phosphate group to it. Okay, following that then, we essentially are going to split our molecule of hexose bisphosphate or glucose phosphate, whatever you want to call it, whatever your spec is. Same thing, pretty much. In fact, in real life, there are loads of little steps, but most of the specs just pick and choose which one they're going to go for. But we take our glucose phosphate and we split it into 2 molecules of triose phosphate. Okay, so we're going to form 2 times TP. Okay, so glucose, as you can see, has 6 carbons. TP, as you can see, has 3 carbons. So I'm splitting it in half, and I'm forming my 2 molecules of triose phosphate. TP, so yes, some of the specs do call this GP as well, which is quite annoying. So depending on what spec you're doing, it might say GP as opposed to triose phosphate. Again, it's frustrating because TP is also sometimes called GALP. So with this, go and have a look at your spec and just see what they're calling these molecules, because some of them do call them different things, which is very frustrating for people like me who are trying to do like all of the specs at the same time. But it just is what it is. GP glycerate phosphate. But anyway, so I'll split it in half. The final 2 steps, and this is where I think it's really fundamentally important that you understand what's going on here, is you need to be able to talk about what happens to TP to turn it into pyruvate. Because these molecules, to me, look very, very similar. They've got the same number of carbons. They're just losing certain things. So the first thing we're going to talk about then is I am going to turn some NAD into reduced NAD. Okay, so remember, the important part of photosynthesis, of respiration, is to make reduced NAD or NADH, because that can ultimately get used at the end. So if I'm reducing NAD, my next step is I need to oxidize TP. So if one thing is reduced, the other thing is oxidized. Okay, so NAD is going to get reduced by having hydrogen removed from it. Okay, so hydrogen is going to be removed. And I'll catch up with your comments because I can see that loads of you are saying stuff. How is it split? No, you don't need to know how it's split. It's just an unstable molecule, so it just splits. How do we refer to this knowledge? How do we refer this knowledge to a question? I'll show you an example of a question in a second. What else are we looking at? In our spec, GP and GALP is 2 different things, so which one is it? In glycolysis, in real life, both glycerate-free phosphate and glyceraldehyde phosphate and glucose phosphate and hexose bisphosphate and hexose biphosphate, they're all involved in this, right? So just look at your spec, look at your book and see what one they use. I can't actually tell you what all of them are because I don't know what spec you're doing. So is this called dehydrogenation? Yes, this is called dehydrogenation. In AQA spec, TP cannot be abbreviated. Okay, if you're saying that, Zainab, I would trust your opinion. Does hexose bisphosphate get split into 2? Yes, it does. Is it a coenzyme? Good point. I'll get to that in a second. Lots of you are saying it. Oh, I've caught up. Right. So this reduced NAD then, this reduced NAD, people saying that this is a coenzyme, they are totally correct. So a coenzyme is just any molecule that can move electrons or protons, in this case as well, from one place to another, right? So a coenzyme, its sole function is just to move molecules from one place to another, and that's what reduced NAD is doing. So it's going to move hydrogens to the cristae later on, so they can be used in oxidative phosphorylation. Final step then is the production of pyruvate. So I'm oxidizing my TP, I'm removing some hydrogen, and I'm also going to remove some phosphate from it. So production of pyruvate will occur by removing phosphate, forming 4 ATPs. Okay, so ultimately then, we need to say that this process is going to make a net production of 2 ATPs, because I've used 2 to begin with and I've produced 4 at the end. So at the end, you have a net of, I've just written 4, 2 ATPs and also 2 NADHs. Okay, so that is the net product as well as pyruvate as well, and they can get used in the next stage. So what else are we saying? You said this is dehydrogenation, but aren't the NAD gaining hydrogens? Yes. So it is my molecule of TP triphosphate, which is being dehydrogenized. So NAD is being reduced, which means that TP is being oxidized and TP is getting dehydrogenated. So basically, don't get yourself too confused with this. If you add hydrogen to something, you are reducing it. If you take hydrogen away, you're oxidizing it. You just need to remember one, then you can just do the opposite for all of the other ones. What is pyruvate? It's just a free carbon molecule. Where do the other pi groups come from? Inorganic phosphate. They're basically just attached to these triose So they're being a bit secretive here because there are actually some other phosphate groups already attached to my triose phosphate, which I just didn't tell you about. So yeah, get in the habit as well, all of you, of when I'm going to use NADH a lot here, but you need to, for your exams, to call it reduced NAD. And then from that moment on, if you put in brackets NADH, it's then mostly fine. It's like with DNA. Everyone knows what DNA stands for, but reduced NADH, they just like you to call it reduced NADH at least once or twice so that they can see what you're talking about, sort of thing. If you had a longer answer question, feel free to call it reduced NAD and then put that in brackets after, and then you can do it from then on. Okay, so that's everything you need for glycolysis. It's fairly straightforward. You make some ATP, you make some NADH and pyruvate, that's it. Following on from that then, I've got another sort of similar process, which is the link reaction. And this is where you get your pyruvate and you turn it into something called acetyl coenzyme A. So by the end, I'm turning it into acetyl coenzyme A. Now, to do this, again, I'm going to be making the same sort of things that I was making before. So I'm going to make some reduced NAD, and I'm also going to be removing some carbon dioxide. Now, this is easy to remember, this carbon dioxide bit, because you guys know that respiration leads to you breathing out carbon dioxide. Right, so this one should be easy to remember. So pyruvate, you're going to be losing a carbon dioxide or a molecule of carbon, and we're also going to be losing some hydrogen, so we're going to be oxidizing pyruvate. Okay, so in terms of my acetate formation, we want to say that this involves decarboxylation, so that's a removing of carbon, and we want to say it involves the oxidation of pyruvate. So dehydrogenation is taking hydrogen off, like D and then hydrogen, just hydrogen. What else are we saying? Pyruvate is actively transported. That is a good point. Yes, I should probably mention that. Does it lose 1 hydrogen or 2? So when you remove hydrogen, we're talking about just a hydrogen, right? So it's an H at the end of my reduced NAD. How does pyruvate get from a matrix from a cytoplasm? Yes, lovely. So sorry, before I go on, this pyruvate, which I had over here, this doesn't just magically end up within the mitochondria. It does actually use active transport in order to get in. So it's going to need to use some ATP, which is why whenever you see theoretical amounts of ATP that get made from this, it's always slightly wrong because you need to use some of it to get it in. And someone else said something else which was clever, and I can't remember who said that. I don't know. So the last bit then of this is there is a second coenzyme. So reduced NAD is a coenzyme, or NAD is a coenzyme, FAD is a coenzyme, and there is something called coenzyme A as well. And coenzyme A is just another molecule which can carry acetate to the correct place. So acetyl coenzyme A production, that's what CoA means, is essentially where coenzyme A will react with acetate to produce acetyl coenzyme A. And then coenzyme A can basically transport acetate to where it needs to go later on. So it can take it to the other molecules involved in the Krebs cycle. So acetyl coenzyme A production, we want to say something along the lines of, I don't know, coenzyme A joins with acetate, forming acetyl CoA. Yeah, and just again, because I forgot to say it, this is now moved into the matrix. I wouldn't worry about it. No, this is something you need to worry about. Coenzyme A it's just a coenzyme. Yeah, you don't need to know any more about it. It's just another one of these coenzymes that can move molecules from one place to another. So to say you don't need to worry about it is probably a bit, I don't know, I wouldn't recommend that, because I've definitely seen exam questions that say, describe the purpose of the coenzymes involved in respiration. And you'd have to talk about coenzyme A. So just see it as like a little bus, like it's a little bus that moves acetate from one place to another. That's all it does. And then it will leave and it will be used elsewhere to move another acetate molecule along. So it is very important. You don't need to know what its structure is or anything like that, but it's just a molecule. Specifically, this is something that people get confused about. It isn't an enzyme, right? So lots of people say that this is an enzyme. It's not. It's a coenzyme, which is a different thing altogether. It's just a molecule that moves things from one place to another. No. You don't need to know its molecularity. That's a word. Nice word, by the way. Okay, we are smashing through this. This is actually going incredibly well so far. I hope you guys are okay with this. So again, all we've really done is we've taken some ATP, or we've added some first, then we've taken some ATP off, we've taken some hydrogen off of glucose to make pyruvate, then we've taken some more hydrogen off, so we've oxidized it further, we've taken some more carbon dioxide off, and we're left with acetate, which we add coenzyme A to. I will have one more little thing and I'll give you an exam question on this. Okay, so final step then, and this sort of looks like a bit of a mess. And I've seen people, I saw a meme the other day and it was like, the Krebs cycle isn't this. The Krebs cycle is knowing the Krebs cycle and then not knowing the Krebs cycle. That's pretty much it. But again, if we take it slowly, you just need to sort of see what's going on. So this is a molecule we had coming from before. And the first thing that happens is coenzyme A is going to leave my acetyl group, forming acetate again, and it will just go back. So it can carry on shuttling. So I don't know if you've ever got like a shuttle bus to like a theme park or something, they just sort of constantly go and do a little circuit. That's what they do. Coenzyme A will just pop up and then bring another acetate and then pop off and bring another acetate, and that's all it does. And just to explain these words here, so citrate, oxaloacetate, again, depending on your spec, you don't need to know all of these. So AQA definitely just referred to this as a 4-carbon compound. EdXLB also just refers to it as a 4-carbon compound. That one is a 6-carbon compound. This one is a 5-carbon compound, right? So depending on what spec you do, they either want you to know those two words or they don't. So make sure you check. I'm assuming some of you are just saying that. Does this occur twice for every glycolysis? Yeah, so for every glucose molecule, this will happen twice because obviously your glucose is split down into your TPs, which turned into 2 pyruvates, which turned into 2 acetates. But anyway, so first thing I want to talk about with this then is I want to talk about just this reaction here. So step 1 is just going to be that my 4-carbon compound is going to react with my 2-carbon compound, which is going to produce a 6-carbon compound. Okay, so first step is 4 carbon molecule plus 2 carbon molecule equals a 6 carbon molecule. And that's all that's going on in that first step, and arguably COA is going to move back. Okay, so yeah, again with this, there are loads of intermediate things, but you don't need to know them. With this pathway, Aisha, I always taught my students this in the past, just 4C, 6C, oh, that's not a C, that's a 2, and 5C. This is all I taught them, and then I got them to remember all the things that come off at all of these gaps, right? That's all I ever wanted them to know. I didn't think they needed to know any more than that, because there's very rarely anything harder than that comes up. So don't get too wound up with this sort of thing. Okay, so following this, I end up making this 6 carbon compound called citrate. With my second step, what things can we say are happening whilst I have a cup of tea or coffee? So what's happening to citrate? What words can we use? What clever things can we say in terms of what happens to citrate next? CO2 is released, so decarboxylation, very, very good. What else? Aisha also smashing it. So decarboxylation is going to happen. Yeah, removal of carbon dioxide, there it is. And then secondly, we can say oxidation of the, I don't know, 6C carbon. Well, I guess it's technically 5 at this point. So oxidation, I'm just going to be lazy. Oxidation 2. Okay, so this is going to produce CO2, more of that stuff, and it's also going to produce some molecules of NADH, specifically 1. Okay, yeah, we could say dehydrogenated as well if you wanted to say that. I'll accept that. Again, check your spec, see what one they use, and just copy them. Okay, cool. How could we describe the process of steps going on here then? So what words can we use for step 3? Good. So oxidation, reduced NAD is being made. So oxidation, we can say is happening again. What else? So reduction of coenzymes, yeah, Aisha, I'm happy with that too. Phosphorylation of ATP. Abid, I'm sorry about that. So, okay, let's go for, does anyone know a better way of saying phosphorylation here? And be careful, oh, you know, there is another decarboxylation. So decarboxylation 2. I get this question a lot, this next one. What is substrate level phosphorylation? So decarboxylation of the 5C carbon. Yes, Jaden, you're spot on. Daniel, I like having you around. This is good. You should come to all my web classes. What is substrate level phosphorylation? Sorry. This is something that people get really confused about, and I don't think it should be that confusing. I'm going to write it down. Who can tell me what this actually is? ATP created using substrate. Yeah, that's basically it. All substrate level phosphorylation is where ATP is made by stripping off a phosphate from another molecule, right? So glycolysis also has substrate level phosphorylation going on too. So this is essentially all of the things that are really going on at this moment. So I'm making some reduced NAD. So this time I've got 1, 2, 3. So 3 times NADH. And remember that this is going to happen twice for every glucose molecule. So I guess technically we could say that that's going to be times 2. I've got 2 molecules of carbon dioxide. Again, it's going to be happening twice for every molecule of glucose. And then this one I haven't actually spoken about, FAD, NADs and FADs. I always told my students to remember that. So remember your NADs and FADs. FAD is basically like NAD, reduced NAD. Apart from it's not quite as good. It can't provide quite as much energy to make quite as much ATP. But I get 1 FAD is always a fad, isn't there? What's the fad at the moment amongst people your age? Fad times 1, but because it's happened twice, times 2. Your brain is the fad. I like it. And then 1 molecule of ATP too. Let's do that there. Right, but because it's going around twice, because I'm actually having 2 of these for every glucose molecule, it's going to be 2 of them. B, substrate level phosphorylation. You strip off a phosphate from my whatever molecule this is, my 4-carbon compound, and you just whack it onto ADP. That's all it is. So it's like a direct way of making ATP as opposed to oxidative. Mr. Pollock? Who's Mr. Pollock? Oi. Yes, sir, I don't know what that means and I'm confused. Don't like it. I'm watching you. Okay, cool. Right, guys, how are we feeling then? Because that's 75%. That's the hard bit of respiration done. What's another way of making direct ATP? Damn, I don't know. Oh, creatine phosphate. There you go. That's the way of making it directly. So removing a phosphate from carbon, not necessarily carbon, just whatever the molecule happens to be. So feasibly carbon, but it will be like, I don't know, the 4-carbon compound. Those of you who are feeling like you are a free, tell me why, as opposed to just telling me that you're totally confused. Tell me what you're confused about and I'll try and help you. So removing phosphate from a carbon. Yeah, I answered that a second ago. 1.5. Okay, I'm fairly happy with that. Right, guys, I've got an exam question. Those of you that are confused, have a go at telling me what's wrong. Everyone else, have a look at this exam question. So mitochondria in muscle cells have more cristae than mitochondria in skin cells. Explain the advantage of mitochondria in muscle cells having more of these cristae. So bear in mind what happens with the cristae. Go from there. Will you do this next year? I don't know. Quite probably, yes. Tom Moy. All the different letters and numbers and arrows. And it is recorded too. Guys, so sorry, what is the advantage of having more cristae? Think in terms of nice biological words that we can use. So what is the difference? Well, not the difference. What is the advantage of a mitochondria looking like this compared to a mitochondria looking like this? Because that's what it's going to look like in a muscle compared to somewhere else. Angela and Humera, you have got some very good words in there. Daniel, you've also got a very good word. So the more folds you have, the more cristae you have, we can say there is a greater surface area to volume ratio. Okay, so mark number 1, there is a greater surface area to volume ratio. Okay, so I've got more of these folds for the size that it is, right? So that's one thing which is really, really beneficial, but you need to be able to tell me a bit more. So why do you need this big surface area? Some of you have said it. I want to see if the rest of you can remember what this is. So yeah, more oxidative phosphorylation, but why? Why can you do more oxidative phosphorylation? Why can you make more ATP? What is there on these membranes which allows you to make more ATP? Why can you do more chemiosmosis? No, there's no more substrate, no more hydrogen. There might be more of an intermembrane or inner membrane space. Good, Jaden, I'll accept that. So if you've got more of these membranes, don't forget these membranes are absolutely embedded with loads of ATP synthase molecules, loads of electron carriers, loads of other things like that. I know we haven't quite got there yet, but you have more of them, which is a benefit. So we can say there are more ATP synthase molecules. That's horrible. You know what I'm saying. More ATP synthase molecules to carry out. So let's go for chemiosmosis or something. Okay. So there's not going to be more respiratory substrates just because there's a greater surface area to volume ratio, right? What's chemiosmosis? I'll get there in a second. That required some of you to know what I'm talking about first. Anyway, substance X. So this is a standard question. Substance X enters a mitochondrion through the cytoplasm. Each molecule of substance X has 3 carbon atoms. What is it? Just Aisha for a moment. Danny, there is no difference. Yes, well done, team. Pyruvate is correct. Yeah, the one which gets actively transported in. So glycolysis is happening out here, remember. The end product of glycolysis is NADH, ATP, and pyruvate. And pyruvate is actively transported into the matrix. Okay, so in the link reaction, substance X is converted to a substance with molecules effectively containing only 2 carbon compounds. Describe what happens in this process. So how can I end up forming a molecule which has 2 carbon compounds? And what are we talking about? What are they talking about in terms of substance? Well, you know what substance X is, pyruvate. So it requires you to know what the link reaction is. So I'm going from pyruvate to something down here, and even saying it doesn't even say what it is. So it's going down to acetyl coenzyme A. Well done, Aisha. Right, so things that are going on then. Where are we? My screen's gone blurry. Oh, sorry about that. Things that are going on then. What are you guys saying? It's decarboxylation. Good. So decarboxylation will occur. The question is asking you to describe as well. So you need to tell me what decarboxylation is. So what is decarboxylation? Good. So CO2 removed. That'll be 1 mark. And then what's the other things that's happening? We can get more than 1 mark here. Yeah, we've done that. Removing of carbon. Combined with COA, yeah, we could say that. So addition of COA, so molecule binds to COA. If you tell me what the end product is, which gives us acetyl coenzyme A, that'll get you a mark on this question too. What other thing happened too? Removal of a hydrogen. Yeah, so removing of hydrogen, that'll get you a mark. Or you could say oxidation, or you could say reduction of NAD, right? Any of those things, that will get you marks too. Final question then. And I don't know where the actual question has gone, but the question basically wants you to tell me how many carbons are in each of these molecules. So that was worth 2. This one, I think, was worth like 4. So could you tell me what is the molecule? What is the first molecule? How many carbons are in my first molecule? Definitely 6. Lovely. So glucose. How many molecules do I have going on which are getting made in glycolysis? So that 6 carbon molecule is turned into what? Good. 2 molecules or something, it's got to be 3, doesn't it? Okay, so that's glycolysis. That's getting turned into pyruvate because it's decarboxylated. Link reaction. What's happening? How many carbons are in this molecule here? Good. This is 2 as well. Lovely. Let's go over here next. How many molecules are in this one on the left? Good. 4. 4 plus 2. I don't know why I just wrote times. 4 plus 2 is going to be what? Good. That's got to be 6. 1 carbon dioxide has just been lost. So what's this one going to be? That one is 5. I think there's got to be some lag because I'm saying something and everyone's saying the wrong answer or something, but now you're all good. We're all caught up. Lovely. Guys, there you go. That is pretty much all you need to know then for the Krebs cycle. So you start with 6, glucose, go down to 3. So that's going to be your TP or GP or whatever your spec calls it. You turn that into pyruvate. That gets turned into acetyl coenzyme A. That reacts with a 4 carbon compound, oxaloacetate. That makes a 6 carbon compound, citrate. And then you turn that into a 5 carbon compound when you lose carbon dioxide. Oof, bam. Right, okay. Next then, oxidative phosphorylation. How many of you How many of you find this quite tricky out of interest? You don't have to call me sir. My name's Ollie. Okay, cool. Right, this looks mental, but it's actually okay. Okay, you've got to bear with me here because this is going to look crazy all of a sudden, right? As you can see. So I think the really important things to be aware of is the name of the sort of process that happens is chemiosmosis. That is something that happens. It's also called oxidative phosphorylation because it happens in the presence of oxygen. But if we start off, we just made a whole load of NADH, didn't we? So if we go back to here, I've made 1 there, 2, 3. I'm doubling all of those because I said I would earlier because there's 2 glucoses. So I've got 6 of them. I've got 2 of my FADs as well. So that's 7, 8 molecules. 9, there's another one there. 10, 11, somewhere over here. There you go. Right. So I've got loads of these NADs or NADHs which are coming in and loads of these FADHs. Okay. Now, essentially what happens is as soon as these molecules get near to the cristae, so this membrane here is the cristae, as soon as they get near, hydrogen gets taken off of them. So hydrogen is going to get removed from my NADH, which is done by an enzyme called a dehydrogenase. Okay, so NADH and FADH are basically going to release their hydrogen, but at the same time, they are also going to release electrons. So NADH and FADH release hydrogen. In fact, let's call them hydrogen ions. Let's get this right. Hydrogen ions and electrons. Now, NADH, and it depends because the science is changing all the time, we typically think that this is about 3 hydrogens here. This is going to, in fact, no, I'm not going to say this till I'm going to change this because this changes all the time. Ultimately, we think that NADH is making about 3 ATPs at the end, and we think that FADH is about 1.5, but this changes pretty much every 6 months. But anyway, first step, I'm breaking NADH down to make NAD. So this can then move back to where it was earlier. So it can carry on breaking down whatever I was just talking about. It can carry on, helping out in the Krebs cycle and in the link reaction. Same with FAD, that can just go back. And my electrons will move into the intermembrane space and they will move down something called the electron transport chain. Okay, so electrons will be moving into the membrane and they'll move down something called the electron transport chain. Okay, now when these electrons move across this sort of chain of proteins, they release a tiny little bit of energy. So they release a tiny little bit of energy as they do it, which allows all of these hydrogen molecules, which I've just made, or hydrogen ions, when I broke down NADH, it allows them to get pumped into the intermembrane space between the cytoplasm and the cristae. Okay, so protons are pumped into intermembrane space as electrons are moved through the electron transport chain. Okay, so I'll say that again. NADH is broken down into electrons and hydrogen. The electrons go through the electron transport chain, and when they do that, that allows hydrogen to get pumped through, right? That sounds crazy because I'm not explaining how it works, and you don't need to know it. So I just accept it that as electrons move through these proteins, for some reason that allows hydrogen to move. Do we need to know the enzyme dehydrogenase? Depending on your spec, yes. Anything else? Anything else? Anything else? No. Cool. Yeah, if you're pumping hydrogen, that is active transport. So this is a form of active transport. Okay. Now, the next thing that happens then, all of these hydrogen ions, which are forming up here, they form something called an electrochemical gradient, right? It's a chemical gradient because there's more on one side than the other. So they want to move down. And it's an electrical gradient because these are all positively charged compared to this side, which will therefore be negatively charged compared to that side. And they don't like that. They don't like being the same charge. So they're going to try and go to the side that they're attracted to. So all these hydrogen ions will move to the other side through a protein called ATP synthase down an electrochemical gradient. and that is referred to as chemiosmosis. So we're going to say that hydrogen, so a hydrogen ion gradient is established. And we're going to say that H plus moves through ATP synthase. And when that happens, like when hydrogen moves through, it causes a conformational change in ATP synthase. It literally starts to spin around, and it causes ADP to react with inorganic phosphate or a phosphate group, making ATP. Right? I know that this is also crazy. Like, why does hydrogen moving through a little protein channel make ATP? It just does. It just causes it to spin, and that makes ATP. It's a really weird, wacky molecule, but that's what happens. Okay. So final thing that happens then is I've now got a whole load of hydrogen ions, which have built up over here because they've just moved through. I've got a whole load of electrons that have just moved through the electron transport chain, the ETC, and they've now ended up down here. So the final thing that happens is I need to kind of get rid of them because I don't want them to really build up. So all these hydrogen ions are going to react with oxygen, as will the electrons. And when they do that, you make water. So that is why you breathe out quite a lot of water, because you're making it in respiration. So oxygen, which is what you breathed in, is reacting with hydrogen ions and electrons to make water. And that's why this is oxidative phosphorylation. Okay, so oxygen is the final electron acceptor reacting with hydrogen ions and electrons, making H2O. Bam. So yeah, that's all oxidative phosphorylation is. So it breaks apart, NADH breaks apart, FADH breaks apart. Electrons move through the electron transport chain, down we go. As they go down, some energy is given over to hydrogen, which gets pumped through. You have a massive concentration of hydrogen ions up here, and then they start to move through ATP synthase, which spins around, making some ATP. So why is oxygen important? Oxygen will react with all of these hydrogen ions and these electrons. So it will react with all of these things, making a product. You guys know that hydrogen is acidic if it's by itself, right? You don't want it to get really acidic in here. So oxygen is going to react with hydrogen and electrons to basically mop them up. That's why oxygen is important. Okay, how are we feeling with that, guys? Let me know if you're a 1, a 2, or a 3. Hopefully you are not this person. Hopefully you are this person who is very happily stood on that one, who also seems to have a bit of an organ. This is good. This looks slightly better than it was before. There were lots of 1s and 2s there. 1.5, yeah, okay, fine. 1.5 can be like somewhat happy. 2.5 can be somewhat sad. 0.1. 0.1 I'm going to accept. 0.1 is like, I don't even know how to draw that. There you go, like the Joker. There you go, bam. Super. Right, okay. So here's a final exam question to have a look at then, and then I'm going to stop here, I think. So the diagram below shows the electron transport chain, which is part of aerobic respiration. So using the information in the diagram, name substance W and explain how it is formed. So what is substance W? You see this question everywhere. Respiration, I would bet my right arm that you'll see a question on respiration in your exams. I'm pretty bold like that. So W is NAD, correct? 1 mark. How is it formed? How is NAD formed? So formed by oxidation. So yeah, okay. So formed by oxidation of NADH. What does that mean? You have to tell me what it means to get your marks. So it loses hydrogen. But what happens to the hydrogen? So I want to be quite specific with hydrogen. So at the moment, I've got hydrogen like that. It loses hydrogen. And the hydrogen atom splits. So it loses hydrogen, forming what then? 2 protons, 2 H plus, and two electrons. Lovely. Right, bear in mind, right, these charges always have to cancel out. So if you've got 2 hydrogens, 2 positives, you need 2 negatives too. Okay, that is all your marks. So 1, 2, and 3. Okay, name substance X and explain the link between the formation of substance X and the hydrogen ions shown on the diagram. So substance X is nice. Do you need to know the numbers of electrons and protons? Yeah, I'd say that's probably useful. So you will see NADH as NADH2 sometimes written. So again, look at your spec, see what they call it, and to go with what they say, as opposed to just guessing, because it's going to be different for different ones. So X is ATP, absolutely. So what is the link between ATP production and hydrogen ions? Aisha, check your spec, see what they say, and then go with that. So movement of hydrogen ions, you need to say a lot more than movement of hydrogen ions. So, okay, so electrons move down electron transport chain. That's creditable. What else can we say? If you literally use the word chemiosmosis, you will get a mark as well. I'm not stopping here though. What else can we say? So electrons move down the electron transport chain. What happens then to hydrogen? So I will say that hydrogen ions move through ATP synthase and ATP is formed, but we're not saying the really important step. Hydrogen diffuses, series of redox reactions for energy for phosphorylation of ADP. Yeah, but what's happening with hydrogen though? I know we've got 3 out of 3, but I need you to be able to tell me what happens with hydrogen if it's going to wind me up too much. Zan, I don't know if I pronounce your name. Zainab, you are totally, absolutely bang on. So electrons move down the carriers, which pumps hydrogen ions to intermembrane space. And you want to use a word like establishing a concentration gradient or a electrochemical gradient or something like that. Lovely. Okay, accumulation of hydrogen ions does make it acidic too, but that's not so important. So ATP is substance X. The association with that and hydrogen is going to get moved through because of the electrons moving through the electron transport chain. They build up in their concentration, so an electrochemical gradient is established. Hydrogen will move through ATP synthase. ATP is made. If you fancy it, you can say that there is a conformational change of shape as well. Something along those lines will get you as many marks as you could feasibly want here. Right, to finish off then, guys, I need to basically sell you something, as I'm sure you guys would expect with anything that's free. In case any of you have not heard of this, and I'm sure some of you have heard of this, basically we are doing some seminars, and look, you can see that someone has gone to the effort to make something that looks vaguely like me. I think there's a picture of me wearing that shirt. There you go. There's me wearing that shirt. And if I scroll down, which I can't do because my thing just broke, there is a picture that someone's just made of me. Isn't that weird? No one told me they were doing that either. I thought that was very bizarre. Yeah, it's going everywhere. So essentially what's going on then, guys, is I am doing some lectures in Imperial College in London. So this is pretty relevant if you live in London, possibly less so if you live in like Edinburgh or something. But I will essentially be going over year 12 and year 13 A-level biology stuff. So I will be doing some sessions on exam technique, for all the proteins. We'll be having lunch, which will be lovely. Cell division, mass transport in animals and plants and various things. Doing a Q&A session. I'll talk about price in a second. I'm doing a year 13 one as well for those of you who are like doing your finals this year, where I'll be going over the harder stuff, so photosynthesis and respiration, inheritance, DNA tech, and basically hard calculations. And yeah, I will basically be there for 2 days doing these things. And before anyone tells me, yes, it is quite expensive, and I can only apologize that it is quite expensive, but it costs an awful lot of money to rent out Imperial College's lecture theatres. And also it's a lot of me talking, and I want that on that day. So for those of you that are interested, you should definitely get on this quite soon, because I am hearing that it is basically selling out pretty quickly, and it always does because it's right in the middle of London and it's just before exams. So I think this is going on as well, because I didn't actually say that, in April. So most of the exams are starting at the end of May, and I think they finish by the 15th of June. I think I'm right in saying that. So this will be just before as like a really quick crash course, well, quick, as a day-long crash course for you to do basically as much as you can. And we also have chemistry, and we also have physics, and we also have maths. So if any of you do any of those, we also have a discount code if you want to do like biology, chemistry, and physics or whatever. So essentially, this is what we're offering. And if any of you are interested, you know who I am, you know what my teaching is like. If any of you are interested in this, feel free to pop along. It'd be really cool to actually see what some of you, I'll see who you actually are. Could you do a video on 25 mark essays? Priya, I would quite like to do that, and I'm thinking about it, but it's totally irrelevant for everyone who doesn't do AQA. Anyway, otherwise, guys, I'm going to finish here, and if any of you have some questions, I will have a go at answering them. So, Bookworm, earlier in the video, when you spoke about how we get reduced NAD, you said hydrogens are removed from it. Just a quick correction, NAD becomes NADH, so its reduction comes from hydrogen being added. I can't remember when I said this, Bookworm, but there's a good chance that you're right, yeah. Anyone else saying anything else? No, I don't think so. Oh, excellent. Right, guys, in which case then, you should now know some things about glycolysis, the link reaction, the Krebs cycle. You should know some things about oxidative phosphorylation, and hopefully you're feeling a little bit more confident with respiration, because I know it's really, really hard. So otherwise, I'm going to call it a day, and I hope you have a really lovely evening. I'll try and answer some more questions too. So Pavati, in terms of yield, totally depends. So it depends what book you use, depends how reliable your spec is, but anywhere from 32 to 36 is what you commonly see in textbooks. So it totally depends. Football highlights, that is very weird. Totally depends on what book you have and if they take into account active transport, if they don't take into account, but theoretically, like about up to 32. Yeah, Daniel, I was going to do it, but I was going to massively go over if I did. So no anaerobic respiration. It's very easy though. It's basically anaerobic respiration, for those of you who are interested, is football highlights. There's no less weird. Anaerobic respiration is essentially where you produce NAD. So you actively try to make some NAD so that glycolysis can continue. And that's pretty much it. Do I do online courses? Yes, I do all the online courses for SnapRevise too. A little bit. Right, does it become charged? It is charged, yeah. There's a massive buildup of hydrogen ions, so it is positively charged in comparison. I am going to finish there, guys. Right, have a lovely evening, guys, and I will catch you later. Tom Moy, yes, I agree. It'd be good if you did understand it a little better. I know it's obviously really hard, but see you later, guys.